Thank you. Can I just quickly ask, has anyone heard of Linguamatics here? Okay. And does anyone know what natural language processing is? Okay, good. So I'm going to try and cover both those things in about 5 minutes, so wish me luck. So Linguamatics, who are we? Well, we're a company that's based both in Cambridge in the UK and Boston in the US. And what we deal with is natural language processing text mining software. And if you don't know what that means, essentially it's about taking text, unstructured text, typically in narrative form inside scientific literature for a lot of our customers, and going in and recognizing the concepts and entities and the relationships between them. And then we can pull out assertions, relationships, and facts from there. Excuse me. So primarily software, but we do some consulting, and we also host a load of content. So we host things like patents, Medline, clinicaltrials.gov, FDA drug labels, NIH grants, a number of other things there. And that's so that we save our customers the sort of hassle of going through and creating the indexes for them. And who are our customers? We're primarily life sciences, pharma and biotech, so most of the big top 20 pharma are using our software. We're also now growing quite rapidly in healthcare. Healthcare has a big requirement for natural language processing. And so we've got sort of cancer institutes, Kaiser Permanente, and we do have some government organizations, most notably the FDA, are using our software internally. Okay, and here's just a sort of like select. These are not all our customers, but some of our customers. And so one of the things I'd like you to take away is that we take a wide range of approaches when we try and analyze the text. We use terminologies, so sometimes these can be referred to as dictionaries, ontologies, but essentially, you know, these are a structured way of classifying types of entities. And so people use things like gene ontologies, diseases, and pharmacological substances, this kind of thing, as ontologies to categorize a whole set of particular types of entity. So you can look for anything that's breast cancer, for instance, or cancer in general. We also use NLP, not neuro-linguistic processing. It's programming. It's natural language processing, and that means that we go in and we recognize the text. We understand that the text is composed of things like nouns, verbs, and any other types of linguistic units within there. And we can use this, a combination of these approaches, along with things like regular expressions, to make sure that we find things in context. And that's the big thing when it comes to chemical structure search, is that we find things in context. And I've mentioned chemical structure search, and that's what I'm going to concentrate on for the rest of the 2 or 3 minutes that I have now. But we use lots and lots of different approaches to find the things, and we can find things in context, and we can apply some disambiguation so we find the right things that people are looking for. Okay, so here's an example, and I apologize for, I don't know why it's in yellow, but essentially here's a piece of text, and just as a sort of like a nice easy way to sort of understand what we do. When our software looks at this text, we're looking for sort of entities. So we notice that is a chemical structure of some kind. And using the ChemAxon technology, which we've integrated into our software, we can recognize that indeed is a chemical entity of some kind, and we can assign a smile to it. We can also identify that COX-2 is a gene. And then because we've got linguistic processing, we can look for words which are basically trying to associate things together. So in this particular case, we can go in there, have a look at the text, and then pull out something which says that nimesulide inhibits the gene COX-2 in some way. And that can be quite important in bioinformatics. And so we can pull out the information. We can pull out the information around the chemical that we found to derive some kind of assertion from there. And we do this at scale. So we do this basically sort of like on millions and millions of documents, tens of millions of documents. So this is just one tiny example. So what do we use ChemAxon for? Well, basically we use ChemAxon to kind of like recognize the chemical structures on the fly. We assign smiles, or alternatively we could use INCHI as well. And then once we've done that, we can pull out the context around the chemical. And so, in the case of, here's an example, we create an index, and typically with something like patents, we will pre-process that, say for instance, some of the mol files within patents, and we'll assign structures to that. And then where those mol files are associated within the patents, they can be put inside the patent documents. A user will go in and query using either a substructure or a similarity search, and then they'll get results out. And I just want to show you a couple of examples. So this is just showing you that you can just draw and search for things using Marvin. But here are some of the examples. And so what we're pulling out here is, for instance, in the top case, over here, you can see that we're pulling out, a particular chemical, and that the fact that there is, in fact, a melting point associated with it. Obviously, if you output it as smiles, you can put it into Excel, and then you can view it as a chemical structure as well. We're very good at doing things like structure-activity relationship extraction, because we can pull out not only the chemicals but the surrounding text around it. So we can pull out some really important information which combines the chemical and bioinformatic information together. And the other important thing is, say, something like in patents, where people deliberately try and obscure things by mentioning one thing in one place, then kind of mentioning it by reference in another place, and then kind of linking it somehow together. You have to read through and try and pull all the information together. We can actually pull that together. So example 12 may be defined somewhere in the document, and then it might be mentioned inside an actual table later on. And when we pull out the results, we can pull all the information out together there. Similarly, the same thing can be used as references to R groups within for Marcuse structures. The definition of the R group may be somewhere in the document, and then the R group may be referenced somewhere else within the document, and then we can pull out all of that and present it in a single row within a table, so that you can understand what R group was being referred to. And what I'd say is, if you step outside of here and look to the right, there should be a banner, and my colleague Andrew Hinton, and we welcome you to come along and say hello to us. Thank you.